Hello and welcome to StudyIQ. I am your friend Rahul Saigaonker. The agenda of today's discussion is connected to biotechnology. Under the biotechnology sector, we are going to specifically talk about genome sequencing. In fact, I have received many messages from aspirants to give clarification regarding the meaning of gene, genome, how genome sequencing happens, what is genome sequencing, what are the advantages of genome sequencing. So I thought it's a good opportunity for us to discuss this, because there is a context too. Recently, the Department of Biotechnology has given a news that our own Genome India project would be completed by the end of 2023. So we will be talking about Genome India project also, then we'll be talking about genome sequencing and all the nitty-gritties which are connected to it. That is going to be the prime agenda for discussion. Let's begin without any delay. But before that, do remember that the admissions for our batch 4 are closing today. 6th May is the last date for you to get admitted into the most comprehensive batch for UPSC civil services preparation, where we will be hand-holding you from your day one. That means from prelims till your interview, StudyIQ will be with you. If you clear your prelims, civil service examination 2024 preliminary, then your mains preparation and its expenses would be completely borne by StudyIQ. You will be called for the mains residential program. You'll be in Delhi, where we'll be guiding you for your mains preparation. There would be mock interviews. There is already one-to-one mentorship available, and all this comes at a very affordable price. And there is discounted price also if you use my code Rahul Live. You can avail this code on the website or on the app. Whenever you check out, just put the coupon code Rahul Live and then apply. You'll get the discounted price. I'll see you in the class. Right, let's talk about genome sequencing. But before that, let's talk about the news. What exactly has happened? Recently, the Department of Biotechnology has given an update over the Genome India project and have said that we are about to complete it. In fact, 2/3 of the project has already been completed. Almost 7,000 samples have been sequenced. 7,000 genomes have been sequenced. And out of that, 3,000 are already available for public for research. Now, we need to understand, what is this Genome India project? What am I talking about? What are these 10,000? What is this 10,000 number? See, in 2020, the Ministry of Science and Technology, it approved a project called as Genome India project. Under the Genome India project, 20 institutions have come together. It is being spearheaded by Department of Biotechnology. Under that, IISc is one of the important institutions, IISc Bengaluru. More than 20 institutions are involved who are going to sequence the genome of 10,000 individuals. The idea here is to take 10,000 samples from Indians from different areas, from northern India, southern India, eastern, northeastern India, and then sequence their genome. This genome sequencing project is going to focus on predictive diagnostic indicators. Focus is on more and more understanding about genetic disorders, understanding about specific lifestyle diseases which are prevalent in India in certain areas, and it is going to be run in different phases. And as I told you, now more than 7,000 genomes have been sequenced, and out of that, almost 1/3 are already available for the researchers to see. Now, even before this Genome India project, which was approved by the Ministry of Science and Technology, it is under the Department of Biotechnology, under this ministry, which is running this. As I told you, even before this, there was a project called as Indigen, which was already being run by Hyderabad-based Center for Cellular and Molecular Biology, where more than 1,000 genomes were already collected. This was clubbed into the Genome India project. And now, by the end of this year, we will be completing our Genome India project, which is going to be a landmark event or a watershed moment for India's research and development in biotechnology. Right? This is basically the update from prelims point of view, where you can expect certain question on what is Genome India project, what is Indigen. You can see some of the pitfalls is 10,000 samples, 10,000 genomes are going to be sequenced from different parts of a country. More than 20 institutions are involved. Of course, it is under Ministry of Science and Technology, but it is spearheaded by the Department of Biotechnology under that ministry. Right? So these are some factual information for you. But the big question here is, sir, you have been mentioning about this genome repeatedly. Genome sequencing, this, that. What exactly is the meaning of genome. And I want to understand the difference between genome and a gene. The basic definition of a genome is the entire set of genetic information that is present in an organism or an individual. That is the meaning of a genome. Means your entire DNA information has to be sequenced. That is the meaning of a genome. And not just that is present in your nucleus, that means in your somatic cell, but which is present in mitochondria also. Remember, we have DNA in our nucleus, we also have DNA in our mitochondria. So entire information, genetic information, when it is mapped, when it is sequenced, it's called genome. All right. Now the question is, sir, how is it different from gene? I want to understand it more clearly. All right. Say, let me explain you what exactly is the difference. See, gene is basically a part of your DNA molecule, whereas genome is your total DNA of the cell. For instance, if I draw the DNA in a very simple way, this is a double strand DNA, and gene is basically a locus on this particular DNA. Say, for example, it's a code where there are nitrogen bases approximately, say, 60 to 100, right? Nitrogen bases, they form one particular code, and this represents a specific trait or a specific character. For instance, this region is going to give you, or this region is going to lead to synthesis of some specific protein or a specific chemical, some specific trait. That is a gene. Gene is basically a locus on your DNA, but genome is this entire thing. Entire information is called genome. All right. So that's the basic understanding between gene and a genome. All right. And there is one more term called as gene mapping, right? Gene mapping. See, genome is the complete set of DNA information. See, for example, if my DNA has this much of information, that becomes a genome. Gene mapping is specifically in that DNA. What does that gene do, right? Meaning, X gene is responsible for synthesis of protein X. Y gene is responsible for synthesis of protein Y. So that is called as gene mapping. So there's a difference between gene mapping and gene sequencing as well. Remember that, right? Right. So genome and gene is done. The next question is, what exactly or how exactly genome sequencing is done? As I told you, genome sequencing means you are going to identify the entire DNA pattern of an individual or of an organism, where you are going to completely sequence the base space, the nitrogen bases that we have. We all know that we have adenine, thymine, cytosine and guanine in our DNA. So once you sequence the entire set of base pairs in our DNA, that becomes your genome. Do not get confused with genome sequencing and DNA fingerprinting. So DNA fingerprinting is basically finding out some microsatellites in your DNA to identify that particular person, right? Genome sequencing is sequencing of all the base pairs of an organism. It can be for humans, it can be for anything. See, recently there was a news I was reading, I think a few months back, last year December if I'm not wrong, that scientists, they mapped or they sequenced all the genes of pomegranate. There would be less number of information available in it. For instance, if I have to map a genome for virus, it would be ending in few kbs. But if we talk about human genome, let's try to understand this. What exactly is the meaning of a genome? See, human genome, it has about 3 billion base pairs. As I told you, it's a double helical structure. DNA is a double helical structure where there are 4 nitrogen bases, and the sequence in our DNA is made up of 3 billion such base pairs or combinations. And these 3 billion base pairs, they basically translate to some genes, about 20 to 25,000 genes, which have already been identified under the Human Genome Project by United States of America. See, what happens in our DNA? In our DNA, there are information, or there is information in these base pairs where, if I say this is a gene, it has some characteristics. Say it produces protein X. There are many areas in our DNA which we do not know what exactly happens. There are areas in our DNA which regulate the working of this gene, which have regulatory functions. In fact, there are many areas in our own DNA which we do not know what exactly is the job of that particular sequence, which is also called dark genome. We know that, right? So your genome sequencing is to understand everything, the complete set. And how is it done? Now you have a question, sir, I understood. Okay, what is genome? For example, a viral genome which is not mutating per se, it is going to be in few kbs. Now if I create my own genome, say imagine if I create my genome means I'm going to build my DNA vertically, you know how much it would reach? It would reach approximately around 60 to 70 stories of that Burj Khalifa, right? Imagine that is the amount of data that is present in our DNA. Now the question is, how do we do it? How do we do it? See, how do we do? We take out a DNA sample. Now I've given an example here of bacterial culture. I take out, say if I'm doing it for human sample, I take out human DNA sample, I extract DNA from it. Once I extract DNA from the chromosomes, what I do is I cut that DNA into millions of parts, millions of pieces. How do I do it? I use restriction enzymes, restriction nucleases. It's called DNA shearing. I cut the DNA into small pieces. And once I cut this DNA into small pieces, I amplify this by the polymerase chain reaction, that is by PCR technique. And once I do a PCR, then I map this in the DNA library sequencing. These days, a lot of equipments have come, a lot of analytical tools have been developed under the domain of biotechnology that I directly get a code like this, a restructured genome. It looks something like this. It's one, this code translates to one side or one strand of my DNA. The corresponding strand can be easily mapped, isn't it? That's the genome sequencing. Now there are different techniques also which we use here, right? Slowly and steadily we have been developing this. Earlier we used to use the qPCR technique, that means quantitative polymerase chain reaction technique. Then slowly and steadily we came up with the Sanger sequencing, right? Frederick Sanger, it's named after the scientist Frederick Sanger. Sanger sequencing. The two most extensive techniques that are used today are Sanger sequencing, and now we are using targeted next generation sequencing. In fact, these days, if anybody talks about genome sequencing, we continuously use the next generation sequencing methodology, which is going to give you the complete picture of your DNA information. So a simple MCQ can come up where qPCR, Sanger sequencing, targeted NGS, what are these? These are genome sequencing techniques. Now the question arises, sir, I understood what is genome, what is genome sequencing, what is gene mapping, what is a gene, how is it different from DNA fingerprinting? What can be the advantages and disadvantages of this? Yes, there are certain disadvantages also. What are the advantages? Now by now I can understand. See, if I'm able to map my own DNA structure, the entire DNA information, what I can understand what causes diseases, I can understand about genetic disorders, I can understand about gene expression. Can I regulate that gene expression? If I study the non-coding part of my DNA, the non-gene part of my DNA, then I understand how a gene is regulated. I can also build a complete base-to-base scan. I can also create personalized medicines, isn't it? Say, for example, my genome is given in the Genome India project. Now, for that particular genome, I can create personalized medicines, possible in future. Or maybe in future, we may assemble some sort of a novel gene, which might not be there, right? That is also a possibility. Apart from that, we can use genome sequencing for vaccine development. We can use it for medicine development to understand the efficacy of the medicines, how exactly they work. We can use it in bioinformatics, of course. Finally, we can also use them to understand rare genetic disorders. In fact, you do not believe a single gene or a change in a single gene in humans can lead to about 10,000 different diseases. So we'll be able to understand what exactly is happening, right? So it has immense possibilities. But there are certain cons also. That means there can be certain potential problems with respect to genome sequencing projects. The first thing is, of course, it's a costly affair. That means you need money, right? The government is going to take care of that. But many a times when you're sequencing your genome, there might be issue that a gene has mutated. It would be very tough for me to understand the difference between mutated and a normal gene. That's a big problem. Apart from that, we do say that yes, we can assess different kind of diseases, genetic disorders, but still screening of all diseases might not be possible because it is going to be huge amount of data. Apart from that, data security is a big concern for a country like India, where there is no data protection act or no data protection provisions. Data security is always a threat. Of course, The government is keeping the samples of 10,000 genomes. Yes, it has been open for researchers, but still, is there a possibility of hacking? Is there a possibility of misuse of this data? Is there a possibility of scientific racial discrimination? Meaning, from one particular area, if I've gathered some samples in that particular genome, I find some vulnerability. Can I weaponize it? There are still issues with that, right? So these are some of the pros and cons. Again, some questions might come up in UPSC mains where you are asked the question, what is the meaning of genome sequencing? What are the advantages? Are there any issues with Genome India project? Are there any issues with genome sequencing? This kind of analysis would be needed for you. Let me just end my discussion with some information for you. Of course, we are running the Genome India project, but United States of America has already completed their human genome project. It was started in 1990s, right? More than $3 billion have been spent, and US declared that the human genome project was completed in April 2003. Apart from this, there are separate genome link projects which are being run. India is going to collect 10,000 genome samples, and we are already done with around 7,000. But in other countries like UK, China, US, they are running special genome projects where they are sequencing at least 1 lakh genomes from their own population. So it's not just India, but many other countries are running genome based projects, right? That's just an update for you, for your information. That's the completion of today's discussion on genome sequencing. I hope it was fruitful for you. If you have liked this video, you can always follow me on this particular ID at @Rahul Sai 222. If you want to communicate with me, if you have any queries, any doubts, complaints, suggestion, feedback, you can always message me on IG. I do respond on Saturdays and Sundays. Thank you for watching this again. Jai Hind.